Okay, excellent. Okay, so today we want to pick up a little bit with chapter 5, and this is talking about linkage or linked genes in eukaryotes and how to map where the genes are on which chromosome. Okay. And I believe we already brought this up at the end of the day before the exam, right? When I talked a little bit about with you that how we know a gene is linked, or one way to, or how it was discovered that genes were linked, I should say, in fruit flies, was that when people started studying mutations, they found that some of the mutations seemed to always travel together. They always showed up in the offspring, even though we would predict they would defy independent assortment. Okay, so we know, or we suspect, we say genes are linked, and remember that means they're on the same chromosome. If those 2 or 3 traits or characteristics sort of defy independent assortment. That's how we can identify or how we start to study this particular problem, right? And again, it was first studied in fruit flies with Thomas Hunt Morgan and one of his students, and then corn, of course, because there was a lot of research being done on corn for agricultural reasons, as well as predominantly, which is a plant, right? So people are like, oh, let's see if Mendel's rules work in corn. And you unfortunately have had firsthand experience in that when counting the different colors and textures of ears of corn. So those are the two, like a lot of the problems that we'll see are either in fruit flies or corn. Okay. Now, we can map not only which characteristics or traits are on the same chromosome, but we can also determine the distance between those traits if we study 3 characteristics together or 3 traits at a time. Make it high def. Oh, please make me look better. There we go. Adjust for. Sorry, I had to adjust my video settings here. There we go. All the words were mirror imaged because I forgot to click on mirror my video, so we wouldn't have been able to read it. Can you go back and watch this video? Yes. So chapter 4 is already up there on Canvas in the first module. We don't have it. Yes, you do. I just did it. You don't see it? All right, maybe I never loaded the page. I'll put it up there. What will happen is it will appear as a page in the module, and when you click on the page, there'll be a box that has a YouTube play button in it, and you can just click the box and it'll start. Okay. Thank you. So if we study 3 traits at the same time, right, we'll be able to actually measure the distance between those traits in a few situations. So we're going to talk about fruit flies. For my example, we're going to go through a fruit fly 3-point, we call it a 3-point cross. And there are some conditions that you need to set up these crosses. Okay. So it's a little different. It's not a tri-hybrid cross, so to speak. Okay. We're using 3 characteristics that we suspect are on the same chromosome. And we are going to study now how often the frequency of crossing over. So it's a technique. It's a way to study this. So basically what you do is you breed flies or corn that are homozygous recessive for the mutations, and then you cross them with a heterozygote male, basically, so that you can see if there's been any crossing over or moving of the characteristics during meiosis, so during gamete formation, that then get expressed in the offspring. Okay, we don't have to set up Punnett squares for this. Okay, it's more about looking at the characteristics, looking at the number of recombinants, and then using those frequencies of recombinants to determine first which allele is in the center, and then calculating the distances between alleles. Okay, so it's kind of a two-step process. So if we look in your book, for example, and we go through, the example that they give is, looks like some fruit flies, and it looks like 3 traits, I think: yellow body, white eyes, echinus, I forget what that is. That's an eye shape. I'm looking to see if they put them in the right order. Oh, he goes on and on and on with method 1, method 2. Did you read that? No? Yes, don't read it. I think I read it a couple times and was just like, I don't even know what he's talking about. So let's start. So remember, when we talk about fruit flies, the notation that we're going to use is the wild type. We're going to do this for wild type. So that would be a yellow body, right? And what was the other one? White eyes. So this would be, oh wait, so this is not yellow body. This is normal body color, wild. This is wild eyes. Not like wild crazy eyes, but wild in the case, this would be red eyes. Wild body, I think, is black to gray. And then what was the third one? Echinus, and that is a shape. So wild eye shape. The mutants are going to be yellow, red, white, and it's kind of like almost like a bar shape, but that's a different mutation. Remember, in order to see the mutation, you need it to be homozygous, right? So we're going to start to put the letters like designated above some lines to indicate the chromosomes. So we call it a test cross because that's how it is. And I'm outside my boundaries. Right? So we would take a homozygous female for the mutations. So we could see them in the phenotype. Okay. For all 3 mutants. So I'm going to put Y, W, E like this. And then I'm going to put a second chromosome, and underneath it, I'm going to put Y, W, E. I'm just going to let you look at that for a minute. Okay. So what I'm trying to indicate here is these are the traits on the chromosome, right? And we have 2 copies. That's the only way we can see this homozygous recessive fly. It's going to have all of the mutants, right? So this fly is going to have a yellow body, white eyes, and the shape of those eyes is echinus, right? I'm going to cross that fly with what we would say a heterozygous male. But males, remember, only have 1 copy. These traits are all in the X, so the males only have 1 copy. right? So a wild type male. And you'll see in your book, they designate the Y chromosome like that. Right? So this is the male fly. And then what we look for is the distribution of these three characteristics in the offspring. Okay. I'm sorry, what? Actually, since I switched to high def, I have a longer boundary now. I'm out to here. So thank you though. I just noticed that myself, and this one's longer too. I can go all the way to there. More board. Which is always good for me. So when you do a fruit fly cross, because we're not going to actually do those with live flies, usually you take 6 males and 6 females that you know are virgins. So you would have captured them right after they pupate into adults. You collect them every few hours, you sex the flies, you separate them. And then you would take 6 males and 6 virgin females, put them together in a new vial with a playground, right? And some food. And in 14 days, you will have about 200 offspring to count. If you do this multiple times, this is why it's a great model organism. If I set up 5 jars, now I have 1,000 flies to count, right? So, and I only need 30 parents of each type, right? So they're small, they occupy a small space, they're easy to take care of. They're large enough that you can look at them with a dissecting microscope and sort them and sex them, right? So they're pretty simple to use as a organism to study genetic traits. Okay, so then, like I said, now we look at the babies, we look at the offspring, and we end up with, and I'm just going to give you the numbers as best I can, and I'll probably make a few mistakes, but just bear with me. So we're going to end up with some offspring that we refer to as non-recombinant or non-crossover. And I'm looking to see if he puts NOC, yeah. NCO, non-crossover, no crossover events. Usually people just call these the parental types, right? The parental phenotypes, so they look like the parents, right? And of the non-crossover types, so meaning that we have some that are all wild type for these 3 traits, and some that show all 3 mutations. So all wild type are going to be black-bodied, red eyes, and normal shape. And the other non-recombinant is going to show all the recessives: yellow body, white eyes, and echinus shape, right? The number observed of these total was 9,444. One set of what we call the single crossovers, and we're going to have 2 of those. One set of the single crossovers, so these flies have only one of the recessive traits expressed. And in this case, it looks like they are yellow-bodied. I wish they would have written out the phenotypes. This is hard for me. So, sorry, wild type bodied. I'm looking for the mutant. So they are wild type body, but they have both of the yellow body. I think these guys are yellow body, but they have wild. The other 2 traits. The other single crossover that's designated is the minus I shape, and they're wild. And then the double crossovers are the one where we have white eyes. Shows up alone. They're white, or they are the other two, yellow. Let me just put the numbers next to these, and then we'll. So the total for these guys is 150. The total for these guys is 400, and the total for these guys is 6. So the data set has to be given to you some relatively similar to this manner, right? And I'm copying this out of your book on page 103. And what I mean by that is they have to give you the characteristics. They are going to label probably the single crossovers, the double crossovers. If they don't, we're going to talk just now, how do you figure that out? And they have to give you the actual observed numbers of flies of each type. Right? Because you're going to need to know the total number of flies that were counted, and then you're going to use those numbers to actually determine those map distances. Now,
is the top number a combination of all wild and all?
Yes, yeah. And the non-crossovers are always the most common phenotype that you see in the offspring. So if you're given a list of just 4 different phenotypes with numbers associated with them, the first thing that you do is you look at that list, and they might not put them in order, like this is nicely in order. The parents on the top, the double crossovers on the bottom. They might just be a scrambled list. The first thing you do is you look at the numbers to determine which ones are the parents, which ones have the parental genotype and thus phenotype. And in this case, you can easily see, like I said, they put them on there in the right order for you. Now, the other important number from this set that we want to look at is the double crossovers, which is always the lowest number of the set. Because the double crossovers tell us, when we compare them to the non-crossovers, that is how we figure out which characteristic is in the middle of the 3. I'll say that a couple more times. Right? By studying the order of the traits, or the amount of offspring that express the parental phenotypes compared to the amount of offspring, the smallest number represents the double crossovers. Meaning when there's been 2 crossovers that happen in prophase 1 of meiosis, so that the trait that was in the middle gets moved from one chromatid to the other. I'm not going to put these characteristics on this example over here. I'm just going to use A's and B's. So for example, if I had 3 wilds and, right, so if that was my heterozygous parent of the set, right? If there was a crossover event where this wild type characteristic left this chromatid, right, and was exchanged for this one, right, I would end up with, right, because it'd be like this, I would end up with A plus plus and plus BC, right? That's a single crossover event. The other single crossover event would be this one. This chromatid keeps wild type, and then crossed over the other characteristic on the other end. Right, and so the complementary one, I guess I could use yellow for that. Probably won't show very much, but. So then this chromatid would have A mutation, B mutation, but then it would pick up the wild because this piece switched. Remember in meiosis? Yeah. Got the sister chromatids, right? And we're changing out this piece for that piece, right? Or this piece for that piece. So the result of this would then be plus C, A mutation, B mutation, wild type. See how those are both one characteristic moved to a different chromatid, switched, was exchanged. That double crossover, the only way I can get whatever trait is in the middle, the only way I can get that to cross over is if I have two crossover events. I'm going to put it here at the bottom. I know it's not the most ideal. So plus, plus, plus. And then my other chromatid is A, B, C, right? So now if there was a crossover event here and a crossover event there, and likewise, my little nub of orange, I'll do it right-handed, right? I'm trying to make them look like X's. I'm not doing a bang up job of it, right? The only way I get B and the wild type mutate, the wild type and the B mutation to flip spaces, because B's in the middle in this example, is if I get crossover twice, right? I have to cut that guy out and move him. And the only way I can move the one in the middle is if I get a crossover event here and then another crossover event post that particular trait. So this is a single crossover. This is a single crossover. This is the double crossover. Okay. So I would end up with plus B plus, right? And then I would end up with A plus C on the other chromatid. Does that make sense? And remember, because our one parent is homozygous recessive, those traits, we're not going to see those traits unless they're in the, unless we have two copies of them. So we're only going to see them when they pair up with this particular allele, this particular chromatid, right? So we don't have to constantly account for all four chromatids. We only need to really account for the ones that are different, right? So like in this non-parental type here, if I was writing it over in this same manner, let's see if I can do that, right? The non-parental, I would write it just like that. I would write Y plus W plus E plus as one chromosome and the other chromosome as YWE, like that. Right? Because this one's homozygous and this is a male, it only has one copy of X, right? So I really only have to look at what's happening between this chromatid and this one. Right? So those are my two parental types. I had one that expressed all wild type because it was heterozygous if it wasn't a male, right? And I have another parent that's homozygous recessive. So they're showing, and they only have those possibilities of alleles. So I really only have to account for that. My advice to you would be always, when you're confronted with one of these problems, the first thing to do is to put the phenotypes and their numbers in an order over here, right? And then be aware that you may have to change the order. Okay, some people can do this really quickly in their heads. Other people need to write it out step by step by step by step. And especially at the beginning, most of you will probably have to write it out. Okay, so how we determine which characteristic is in the middle, again, is we compare our parental types, which in this example were these. We compare the number of parental phenotypes with the number of double crossovers, which again, remember, is always the smallest set. Okay, so I'm going to erase my example and we'll go back to using our actual numbers here, right? So our parental types are Y, W, E, so wild and recessive, right? Our double crossovers, the ones that we have the fewest of, we have a flies that have white eyes. So we have the white eye trait all by itself. And we have yellow highness together. All right, and we only have 6 of those. So we look at the phenotype and we look for the trait that is all by itself, because that indicates that was the trait that was in the middle that got double crossed out to appear alone. Okay, so in this example, did I do that wrong? Are they yellow, not white? Did I mess that up? Are you guys following along? I think I messed that up. Wild, no, with white eyes. Where we got the correct order? I think I got the wrong. Oh, I'm sorry. I think I got the wrong double crossover phenotype. So no, it's wild with white eyes and yellow highness with white. Oh, they're showing. Okay, got it. So you are fortunate in this example because they put them in the correct order when they gave you the problem. A lot of times it won't be in the correct order. So which of the characteristics is all by itself in the phenotypes of the double crossovers? The trait for the white eyes. What that tells us then is that white eyes is in the middle of yellow and echinus. That's how you find it. You have to have the parents. You have to know which are the parents, the non-recombinant, and you know that based on the largest number of offspring are the non-recombinant parents. And you are comparing that to the least, the double crossovers, and you're always looking for which mutation is expressed alone, because that means it's wild type at either side. And that mutation then will be missing from the others, the other set, the other parts of that offspring, because this would be wild for white in the middle, right? So that essentially means that white eyes is in the middle and it was the double crossover event like this, right? Two crossovers get white flipped so that we end up with wild white eyes, wild or echinus, and yellow body, normal eyes, red eyes. Right? It takes practice. Just write this down for now. I'm going to give you another problem and try to do it yourself. Okay? The math is the easy part. Once you figure out which characteristic is in the middle, you're home free, right? And it does not matter which single crossover you choose as the first one you're going to look at. Usually in a problem, people put the higher number first. Your book did not. That's fine too. It doesn't matter. It's 3 traits on a chromosome. We're looking for the order they are in relative to each other, right? If I'm looking at this chromosome in this direction, Y, white, E, right? It doesn't matter if I'm looking at it in this direction or this direction, it's still the same distance between between those characteristics. So it doesn't matter which one you choose as your first single crossover. That doesn't matter. People sometimes, get fixated on that. We need to find out which are the parental alleles, which are the double crossovers. That's going to tell us who's in the middle, which mutation is in the middle, and then we're going to be able to work the math. Oftentimes, it will mean you have to rewrite your symbols if you have to switch. Like if it wasn't white in the middle, if it was E in the middle, then you would rewrite these. I usually rewrite them with the correct order because then it helps me remember their phenotypes and what their genotypes are. The math is super simple. All you do is you take the number of your first single crossovers, 150, plus the double crossovers, 6, and you divide it by the total number of flies that were counted. Your book already calculated it in a percentage for you, right? So the first single, the 150 plus the 6 tells us the distance between yellow and white. So it's going to be 156 over the total number of flies, which I don't see written out. You can do the math. 1,000. 10,000 something. Is it 10,000 even? Okay. They made it easy, right? And then I think you're going to multiply that by what number do you get there? You do that. Oh, we have to actually do some math. Yes, you have to do some math. So 156 divided by 10,000.
0.0156.
0.0156. And then we're going to multiply that by 100 to give us 1.56. That's our distance. That's our map units between Y and W. And then our other single crossover would be between white and our other trait. And that is 400, right? Plus 6 over 10,000, right, times 100. And I believe that answer is, I'll just tell you what that answer is because it's in your book, 4.06. So then you can draw a map. And literally all you do is you put, like, you go like this, right? And you put white eyes here, and you say, okay, white to yellow, this distance equals 1.56, and white to Eukaryotic is 4, so I'm going to make it about 4 times that size, 4.06, and then you're going to say the total distance between yellow and Eukaryotic, you're just going to add them together. 1.56 plus 4.06, that total distance should be 5.62. Done. Usually they're called map units. People put MU at the end of it. It's not important to me if you do that at all. So I'm going to give you one to try for yourselves. Okay. I'll try to find an easy one, but one that is oh, I like this one. put it up on the I can. All right, I'm gonna erase this so that we can put up some new data. We're gonna try one. I think this is in P's, so it won't be quite as foreign because the characteristics might be a little more familiar to you. I can get it to project. I know you can't see it. I'll make it bigger. I'd like you to take a look at number 20 on this paper. There, can you read it now? That's about as big as I can make it. Three recessive traits in garden peas are as follow: yellow pods recessive to green, bluish green seedlings recessive to green, creeper to normal. Okay, so like a laying down one opposed to a standing up one. A true breeding normal plant with green pods, green seedlings. So true breeding normal plant, meaning normal in ability to stand up, was crossed with a creeper with yellow pods and bluish green seedlings. Okay, so all the recessive, all the mutations together, crossed with a wild type, test cross. Right, so we have a heterozygote crossed with a F1 plants were then crossed to creepers with yellow pods and bluish green seedlings. The following results were obtained. And the reason I picked this one is because, in part, because the numbers are not in order, they're not in sets, and it asks you to do all these things. We're going to double check which trait is in the middle, and we're going to calculate the map distances between these. Okay. You're going to need your lab partners. All right. So I would start with, let's just start with the obvious. I'll help you get started, and then I'm going to leave you to try to figure it out. Okay, three recessive traits: yellow, green, bluish greens. Yellow is recessive to green, bluish green seedlings recessive to green, and creeper. Those are all recessive. So just so we're all using the same symbols, right? Yellow pods are recessive to green. Bluish green seedlings are recessive to green seedlings. So I can't use G again, right? So let's use little b's for blue. And a big B for a green seedling. And the creeper, you want to use C for creeper. So the normal, this would be normal. That would be creeper. Right, so we're going to start with a true breeding of each, cross them together, get F1s. So they'll be heterozygous, right? And then we're going to cross those, we'll do a test cross. We're going to cross those with a homozygous recessive and look at the offspring that we get, right? Normally, if these were not linked traits, we would have that giant box of 64 and we would get various combinations, right? Of so many that were had both of the dominant traits and so many with all the recessive traits, right? But that doesn't happen. You only get these 8 sets of offspring, which is your first clue that the genes must be linked because they're moving together and they're always together. They're not independently assorting. If they were independently assorting, I would have 8 different phenotypes and I would get those standard ratios of the 64s, right? Because I don't, that's one way, that's how we know that they're linked genes. They're on the same chromosome. So let's identify what the chromosomes would look like if I had a true breeder for green, normal, and I crossed that with all of the homozygous recessive, yellow, bluish green creeper, right? You guys can see now that my F1 would be heterozygous. Right? I don't have to make a box of 64 squares or do forked line approach for that, right? You see that all the possibilities, the only possibilities in all of the gametes are going to be this one crossed with that one, right? That's what's going to end up in all the offspring because we only have big G's and little G's. Yeah. Right? Just like if we did a monohybrid cross or a dihybrid cross or another regular trihybrid cross. So they're all heterozygotes, right? So that's the F1 plants. A true breeding normal was crossed with a true breeding with all the mutations. The F1 plants were then crossed to creepers with yellow pods and bluish green seedlings. So all 3 mutants again, all 3 mutations. It's a test, just like a test cross, like the homozygous recessive. So we're going to cross them again with this guy. And these are the results. This is what they look like. And this is how many of each, right? So we're going to put them in sets to figure out which one's in the middle based on which one is the double crossover. But to know which one's the double crossover, we have to know what we have to know which are showing our parents and which one is showing the double then. So if you are thinking about this in terms of the actual chromosomes, right? Like we were doing before, right? That's the heterozygote. Okay, so take a minute. What I would do first, if I were you, is I would take the numbers and put the ones that are the closest to each other in sets. For example, green pods, green seedlings, normal, with yellow pods, bluish green seedlings, creeper. I would put those together and I would put the genetic symbols for their genotypes. I would write out the phenotype first. Write out the phenotype, the symbols that you would put on their chromosomes, and then the numbers as a set. And start with the numbers that are the biggest. Then with the next two sets of numbers that are the closest to each other. So we have 282 and 281. I would put those together. They're probably a single crossover. And then we have 151 and 157. That's the other single crossover. And then we have 15 and 11. So that must be the doubles. So I would put them in that order. And I will give you a minute to do so. Oh, sorry. Yeah, try to do it now. See, here, there. I wonder if I only give you half the numbers. Sorry, I was just. I moved my thing. There. The challenge for me is I can't write on the board and, well, maybe I can make this white. Tell you what, I will help you out and I will make some copies of this so that you can have a copy of it yourself. How about that? Yeah. I'll be right back. While you guys are working. So we have green pods, green seedlings, normal, 2059. Right? Green, normal. Right, so another way to depict that would be green. We were using blue, right? That, you're doing it like fruit fly way, okay? Or if you want to just use the 3 capitals, I think that's okay too. We use for normal, big C. And the other parent is obviously the one expressing all the recessives. So that's going to be little g, little b, little n, right? So there are my 2 chromosomes. And how many of those are there? 2041. These are the non-crossover. Those are the parents, the non-recombinants. Parents, phenotype, whatever you want to call them, right? And we don't need their numbers for anything except the total. In the example that we did from the textbook, they had already put those 2 numbers together and just reported it as a single number. Okay, like I said, we don't need those numbers for anything except figuring out who's the parents, which we already suspected because we had done this over here, right? And then we just need them for the total number of pea plants that were counted. Okay, so let's look at the next. I'm just going to do the next one on the list. Green pods, green seedlings, but creeper. So green pods, green seedlings. Well, it's not N, it's C. Sorry. But creeper. No, actually, no, let's not do that. Sorry. Let's look for the double crossovers so we can figure out who's in the middle before we make any more of our chart. So which 2 phenotypes are the smallest numbers? 15 and 11, and 15, so I'm going to try to put them down here. 15 is green pods, bluish green creeper, and 11 is yellow pods, green seedlings, and normal, right? Normal height. Which one's which? This one's 11, and this one is 15. So now when I look at, and no, you can't see that. So the pluses is just, so that's just because it's Drosophila. Like I'm thinking most of these crosses are Drosophila crosses. So that's the notation you're going to see most of the time. So that, yeah, that's fine. We can use the letters. It's just habit for me not to use the letters, but to use the pluses, because that helps you a little bit with what the phenotype is. You're looking for a wild type phenotype, which is dominant phenotype, right? And then you don't have to keep thinking, oh, is the G for green or is the G for yellow? Is the N for normal? Is the C for normal? If you just see the pluses, it actually simplifies it a little bit. So if you're looking at the parents, wild, 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 green, green, and normal, and the other parent would have been yellow, blue, green, creeper. All right, if you're looking at the parents compared to the ones that are in the lowest frequency, which are the double crossovers, which characteristic is all by itself? Color, pod color, color of seedling, color of pod, or standing up? Pod color, right? This one's all by itself. So what does that tell you about which characteristic is in the middle? Pod color is in the middle. That's what it tells you. How do we know that? Because the only way we can get G by itself, the mutation of pod color alone, our parents, the pod color for green, right? Little g was always with bluish green and creeping, and now it's not. It's all by itself. So it had to be crossed out, double crossed out away from the other two mutants. And the only way that can happen is if G's in the middle, pod color's in the middle. So now what people normally do is they reorder the traits on their chart so that they're in the right order when you go to put them on the map at the end. Okay, so all I'm going to do is this. I'm going to rewrite my chart now, and I'm going to put it like this. I'm going to put, it'll still be plus, plus, normal. The parents are, but I'm going to put the G in the middle this time. Bluish green, yellow pods, and creeper, right? Yeah. Because this was green. No, this was yellow. Why did I choose that? Oh, yeah, this was green. So this was wild, blue, creep, right? With yellow alone. Yellow, normal, normal. So the only way I can get these two to swap so that I can end up with wild type for the seedling, green for the pod, and wild type for the standing up is if G's in the middle and I swap them out. So then that gives me the yellow alone. Right? Then I would have this one with this one. Okay. So I would rewrite them. And same down here, I would rewrite them so that be B plus C and plus G plus in the middle. And if it helps with the chromosomes, like with the lines for the chromosomes, by all means do it. And so then now we just need to look at our other sets, right? So if let's look for the one that is bluish green seedlings with green pods and normal. Oh, it doesn't really matter. I'm just going to look down the list, right? So if I have green pods, green seedlings, creeper, green pods, green seedlings, that's normal, creeper, That one is 151. And its complement is yellow pods, which is my G, right? Bluish green seedling. Wait, yellow pods, bluish green seedlings, normal, right? And that one has how many? 157. So I'll put the lines in there. And then the last one would be the bluish green seedlings, little b, green pods, which is the dominant, normal, with yellow pods, green seedlings, creeper. What do I have? 281 and 282. And again, it doesn't matter which one of these sets you put first, if you put the lower number first or the higher number first, or if you put them in the other order, it doesn't matter. It really doesn't. So now we can do our math and figure out the distance between these traits, right? So I'm going to take the distance between the pod color and the seedling color, and the pod color first, because that corresponds to these numbers, and I happen to put them on the top of my list. So what I'm looking at is what is the probability, or how often does this happen, right? How often does this happen, this first crossover? Break these guys because they're backwards now. What do we want? Plus. So I'm looking at this crossover first, right? This guy with those two, and this guy with those two. That's my first single crossover. So I'm going to add these numbers together. 308. And I always add in the double crossovers because there was this crossover happening. I'm counting this crossover happening as part of the double. So we add the doubles in each time with the singles. So we add, what's 11, 26. And it's usually some nice round number. What is it? 5,000. How many? 2,500. What's up?
It's 4997.
4997. I bet they really meant that to be 5,000, but that's okay. Usually in the hypothetical problems, you know, they're even numbers. All right, so when you add that together and divide by 4,997 times 100, what do you get?
6.684.
6.684. Can I just say 6.7? 6.684. Yeah, let's round that up to 6.7. And then what is the distance between my other single crossover where I am saying plus, and I'm picking up the C, right? I'm going to put those in the wrong spaces. Yeah, I did. Sorry about that. What's the distance between the other crossover? Let's just do the math. 563 plus 26, 7, 100. How many? 12.79. Can I make it 12.8? 79. So that I can draw my map, right? And put the distances in. I know my total distance between the two on the outside is going to be 12.8 plus 6.7. It's approximate. It's not perfect. right? So it'll be 19.5 for the whole thing. And we know green's in the middle. What is my distance between,
Sorry, it was 11.8.
okay, you're fired from being my mathematician then. All right, so now I'm gonna change it to 18.5. I was counting on you. All right, at least you admitted your error. So total distance is 18.5. What's the distance between, and we already know that yellow's in the middle, right? So what's the distance between blue seedlings and the yellow? The distance between B and G. That's the 11.8. Oh, wait a minute. Did I put those numbers together incorrectly? No, that's okay. Yes, B to G, yes. The blue, green to yellow, that's the big one. So that's 11.8. And then I'll make it a little longer. This will be 6.7. So I'll put it so it looks a little bit skewed. Right? I'll put 6.7 distance from here to here, 6.7. Done. Why do we bother to do this? No one does anymore. That's the answer. This is just an exercise in logic for you, right? This was used extensively, especially in organisms with low numbers of chromosomes, like Drosophila, corn, onions, peas, C. albicans, C. elegans, the nematode. Once you get to organisms that have like 23 different chromosomes, that would be you, like humans, this is next to impossible to find 3 traits that you know or suspect are on the same chromosome because there's so many characteristics. It's almost impossible unless you're studying a particular genetic mutation, and you wouldn't know what other characteristics travel with that unless you had a pedigree of maybe 10,000 people or 10,000 people in families in order to be able to determine the frequency of those genes traveling together, right? So it's totally not practical once you get to be an organism with more chromosomes. So again, why do we bother? One, because this was the way that people first identified a particular gene on a particular chromosome. And that is important still to this day, to be able to locate where in the genome a particular trait resides. All right, so that's one reason why you have to learn this. And as I said, it's mostly historical, right? To illustrate where each gene is located. And again, you can only do this when you have a small or a low number of chromosomes. And this idea to us in this year, right? Easily, we're like, yeah, genes are on chromosomes. But in 1920, people weren't sure.
So the difference between B and G and then
6.7, right? It should, does it equal 18.5? It should equal 18.5. And again, these are in real life approximates, and I'll explain to you why later, right? Because there's interference. Once there's one crossover event, it's less likely that that chromosome is actually going to cross over again. And so the number of observed double crossovers is actually always less than the number of expected double crossovers. But again, we're not crossing fruit flies, so it's not that important to us. You don't need to know that. Okay? Right, so it is important to know where that each gene is located on a particular chromosome. Now, in humans, because of course, whenever scientists do something in peas or corn or fruit flies, then they're like, oh, how does this apply to us, right? People tried to do this and it was next to impossible, like I said, unless you had a huge pedigree. And so later, much later, I'll give you the year in a second here, since I look it up, so I don't remember. This guy devised this really cool method in the 1960s called somatic cell hybridization. If I can even find his name. Georges Varsky. Somatic cell. And so basically what he did was he put some cells together and fused the nuclei, which is not really very difficult to do. You can add a chemical and it causes the nuclei of cells to fuse together. And then what happens as you culture those cells over time, the nuclei, that new giant nucleus, it starts to lose some of the chromosomes. And at first he was just doing it with a single species, but when he saw that the chromosome, some of the chromosomes were eventually being kicked out of the nucleus, he was wondering, well, I wonder if I could do this with two species and use it to figure out where on which chromosome a particular characteristic was located. So he fused mouse and some human cells in culture, in tissue culture, we call it. But it's not really tissues, it's just cells. I don't know why we call it tissue culture. And those cells were named heterokaryons because they were two species, right? Hetero, different species. He made these heterokaryon cells. And like I said, eventually the nucleus fuses, and they call it a synkaryon. No one uses that word. And then that fused nucleus starts to kick out chromosomes. And the ones that are particularly useful are the ones that are going to kick out some of the human chromosomes, right? So we have these mouse chromosomes, and we don't have all 23 human chromosomes anymore. Now maybe we only have 6, or we have all of them except chromosome 2. And literally what scientists did was they tried to build a panel or a set of these heterokaryons. Ideally, that was each cell was only missing one human chromosome. See where this is going? Right? So if I have a set, and in this set, this cell A is missing chromosome 1, heterokaryon B is missing chromosome 5, heterokaryon C is only missing chromosome 7, or only has chromosome 7. It can work either way. And I asked, I think the gene to make this enzyme is on chromosome 1. Let's see which of my sets can still make that enzyme. And if you have a big enough set with only, and you know which chromosomes are missing, which you can do by looking at them with doing a karyotype, you can start to figure out which chromosome has which gene on it, makes which protein. So let me show you a picture, it'll help. It was not a perfect system
Why did they have to use with mouse cells to do that?
because we can grow mouse cells in culture really easily because they were there, right? When you grow human cells, they just keep all their chromosomes unless they're a tumor cell and they might kick one or two out, but that's not interesting, right? So specifically, when he first was fusing mouse cells with mouse cells and started to realize that after the fusion, when you grow them in culture, they don't keep all their, they don't have two complete sets of chromosomes, right? That gave him the idea that, oh, well, what if I fuse them with a human cell? Are they going to kick out the mouse or the human? And that would be a useful tool, right? So let's take a look at this. And even this is outdated. People don't do this anymore. No, we just use molecular biology. But yes, we're waiting too. Okay. I'll make it big as I can. Okay. Okay. So the hybrid cell lines are labeled over there on the far left as cell line number 23, number 34, number 41. And it's showing you which human chromosomes are still present in those cell lines. And you'll notice that line 23 still has chromosome 1, 2, 3, 4, the biggest chromosomes. Line 41 doesn't have chromosome 2 or 4, but it has 7, right? So you're going to get all these different mosaic cells. And obviously it would take a long time to figure out which cell line still had which human chromosomes in it, right? This is what is interesting over here, okay? Gene products expressed. So we would be testing usually for a protein to be made in these cell lines, which is a whole other process and is various depending on what kind of protein we're looking for, right? And what this allows you to do then is to figure out which cell line still has the chromosome that's making the protein. And then if you can narrow it down further, you can figure out which chromosome actually has the gene for that protein on it. And then you can go searching for it specifically. So for example, notice cell line 23 does not produce protein A or C. All right. Let's focus on A. Does not produce A. The other two cell lines do. So which chromosome is cell line 23 lacking that those other two cell lines have? 5, right? Both of them have 5. It can't be 6 or 7 because both of those cell lines don't have 6 and 7, right? So this guy these two cell lines have 5 and cell line 23 does not. So chromosome 5 is a really good candidate for gene A. If we know the protein sequence for gene A, we know it's alanine, alanine, glycine, glycine, blah, blah, blah. We can infer what the messenger RNA would be and then use that messenger RNA as a probe to go looking on just on chromosome 5. Where on chromosome 5 does my probe bind? And then I can start to figure out where on chromosome 5 that gene might be located. Okay. Let's look at another one. If we look at the product B, right? What is missing from cell line 34 that's in common with 23 and 41? Well, 23 and 41 share chromosome 1, right? Only thing they share. But so does 34. So it's probably not on chromosome 1. What other chromosome do 23 and 41 share? 3. And this one does not have chromosome 3. So again, chromosome 3 would be a good candidate for that gene. And we would go fishing and looking for that particular gene on that particular chromosome. You can sense perhaps how laborious this is, right? So you would have to make these cell lines, which a lot of times doesn't work. And then you have to grow them over multiple generations and wait for them to stabilize and then test which human chromosomes they've kicked out and which ones they still have. And then you would freeze them and you would make more cell lines. And then when you wanted to actually go looking for your gene product, you'd thaw them all, make sure that they all still have the human chromosomes that you thought they have. And then you would start doing these tests on these cells. Like I said, you'd be trying to purify a protein from these cells. So you'd have to grow them up in big quantities and try to get them to make this protein, which whatever that's going to take. And then you could finally maybe find out where that gene was located on the chromosome. So a long time ago, when I was a graduate student, and I had to go to these seminars every week of visiting people that would come and speak to us. Unfortunately, although I was studying immunology, my department had just merged with biochemistry and molecular genetics. And all the new people, all the young guys were molecular geneticists. So most of the people that came to give seminars were their friends, right? So and this sticks in my mind to this day. I'll never forget this seminar. We were all sitting there. This woman, everyone's like, oh, it's a big deal. It's a big deal. She's from California. She's from California. She's coming from UCLA. I'm like, oh, okay. Because we were in Ohio. That was like, California is really far away. And why would this person ever want to come to Ohio, right? Cincinnati. So she comes walking out and she puts up her first slide and she says, we're starting a human genome project and I've been assigned chromosome 2. That's like the second biggest chromosome, right? And the map for chromosome 2 had like 2 things on it. They had mapped 2 genes to the whole chromosome. And she proceeded to show those 2 things. And then she started to try to tell us how her lab was going to use that to improve the map of chromosome 2 and what she was doing, right? The 2 things that were on the map, they knew from this. And she was getting ready to try this whole other thing called restriction fragment polymorphisms, which is what your book says people use now to actually map things on chromosomes, right? But it was quite striking because I was like, oh, that's so exciting. Yeah, I'm interested in that, even though that's not my thing. I was like, I've heard about this human genome project. Of course, now it's finished, right? That was a long time ago. But when she stood up there and said, I've been assigned chromosome 2, I was like, it was inspiring. I was just like, wow, that's ambitious. She's going to get a lot of grant money, right? I was thinking all these things. And then she flashed up the picture of the chromosome and there were like 2 markers on it. And I was just like, okay, so what's on chromosome 2? And she's like, well, we don't know yet, right? You will see, I think in chapter 1 in your book, I don't know if you guys remember, but I remember putting this picture up when we started this discussion of what is genetics. There was a picture in your book of some of this one, right? So this is probably a little more relevant to you now, right? Less than 30 genetic diseases, genetic conditions that are definitively mapped to specific locations on specific chromosomes, right? Even 35 years later. Let's see, is there anything on chromosome 2? Ah, there is. What's on chromosome 2? Ehlers-Danlos syndrome, right? Super rare. All right. So just that. And then I want to talk to you about RFLPs for a second, and then sequence maps. And people use a single nucleotide. It's off. Okay, so in the 1910s, 1920s, people started mapping characteristics to particular chromosomes and mapping those distances using crosses, right? Using almost like traditional Mendelian genetics. Today, right, that we advanced the somatic cell hybridization, and now in modern times, people use RFLPs, single microsatellites, I think they call them microsatellites. Also known as short tandem repeats. And then last but not least, single nucleotide polymorphisms. And I know we haven't talked a lot about DNA specifically, but now people use specific DNA markers. These would be considered different DNA markers that are used to, you put a marker down on the chromosome and then you start to walk one direction or the other from that marker, looking for more variations, okay? So an RFLP is a short abbreviation for something called a restriction fragment length polymorphism. Okay, so what that means in brief is if I took all the DNA from one of your cells and all the DNA from one of my cells, and I cut them with a handful of restriction enzymes, which are enzymes that cut DNA into pieces at specific base pairs, right? And I separated all that DNA electrophoretically, I would get a banding pattern for me and a banding pattern for you. And then I would do it with someone else and someone else and someone else. And I would look for what pattern, what band sizes we have in common, right? And then let's say I have a disease that is degenerative eye disease. Then we would look at the 10 and we would look at mine and we would say, what's different about these fragment pieces? Is there any fragment of hers that is smaller or larger significantly than the rest of the population? All right. So in order to do this, you do have to have DNA collected from a large group of individuals, and you usually are comparing a diseased person or a person with a genetic disorder to what we would call normal. There's of course going to be some variation in the normal, but we're looking for a dramatic difference in these restriction fragment lengths. I believe it's a relatively small difference. I think you can tell on a sequencing gel of about maybe 500 to 1,000 base pair difference. So then you would know even closer where to look in that particular person to find where the gene is for that characteristic, right? That's like 1980s technology. Now, because the human genome has been sequenced, you can look for smaller pieces, not these big restriction fragments created by restriction enzymes that might be 80,000 bases long. Now you can actually look for short tandem repeats. You can sequence a person and you can look for these repeated sequences. They're very small, like how many CAs are in the middle of this particular gene. And it's actually different between all of us. And it's most different between people with this particular condition or this particular trait. And of course, you can look even further at a single nucleotide change. I'm thinking of hemoglobin and sickle trait because that's one that's familiar to you, right? So that's one single nucleotide change that changes the protein that changes the shape of the red blood cell that causes sickle trait, right? So this is more where we are now, right? Like I said, this is more like the 1980s and 90s before the Human Genome Project was finished. That was finished about 2003, 2005, and now we can use these things to actually pinpoint even closer. And like your book says, even more than that, they've created these giant databases of sequences, and you can actually just send a computer program hunting for the differences, right? We call that bioinformatics. So we would have these large databases containing DNA sequences for everything that we know. And like I said, then you can ask a software program to go find similarities between all the people that were living in Elizabeth, New Jersey when this particular, toxic thing was dumped, right? Or you can ask it to find all the people that were descendants of this particular group of individuals that lived on an island, right? And then you can get information that way from a really large gene pool. So most of the time when you are, like when you do that stuff, right? Like 23andMe or whatever, right? You're contributing your DNA to the pool, right? To the databases that then are going to be used eventually for, not that they could pinpoint it essentially, maybe or maybe not back to you individually, but your DNA becomes part of the database that can then be searched for various things, right? All right, that's enough of that. Let's have some lab, shall we?